Genomics is transforming how we diagnose, treat, and prevent disease, and is making precision medicine possible. However, the current barriers between discovery research and clinical systems is hampering our ability to use genomes generated in the clinic for discovery research and slowing down our progress towards precision medicine. This is because research and clinical systems have very different requirements. Clinical grade system requires stringent quality control such as ISO standards, robust auditable infrastructure, and need to adhere to health privacy laws. Research systems, on the other hand, do not need to invest into these costly measures. Instead, they require a managed process for accessing raw data, catering to consent changes, and enable unrestricted research workflows at scale. Therefore, clinically generated data remains fragmented across disconnected systems, limiting clinical insights and knowledge discovery. TRECA bridges clinical genomics and discovery research by managing both through one system, enabling clinical workflows and reporting, then transitioning the data into a secure research environment with role-based and granular access to individual files or cohorts. TRECA is built with interoperability at its core. It adheres to cybersecurity standards and is fully auditable, being built in accordance with ISO 9001. It caters to real-world clinical workflows, such as rare genetic disease and pharmacogenomics processes, which include the annotation of genomic data from clinical databases, prioritizing variants according to custom rules, and enabling clinicians to add notes or questions ahead of a multidisciplinary teams meeting, as well as compiling everything automatically into a clinical report. TRECA also caters to the scale, complexity, and freedom needed in modern bioinformatics research. It enables researchers to spin up secure compute environments and ingest data that they are cleared to access. This can be command line, notebooks, or Nextflow pipelines with software freely installable, and data export under policy. Adhering to standards and catering for real-world flexibility, TRECA can hence interface with other trusted research environments such as UK Biobank. Let's look at the system in action. We follow Jill and Pam as they create a clinical report for a cystic fibrosis patient. Jill is the clinician and needs to run the clinical workflow, review the variants the system discovered, and flag the right ones for Pam to review in her capacity as medical director. Pam reviews, and when satisfied with the supporting evidence, approves the variants for clinical reporting and generates the diagnostic report. After Jill logs into the system, she can go to the Clinical tab to submit a workflow on a specific patient file that she has access to. She needs to give the job a name and submit. When completed, she can see the outcome in the Clinical Workflow Results tab. Here, she can first review the analysis workflow and what decisions the system made along the way. When satisfied, she can review the variants found either by going through the list or using filtering criteria to navigate to pathogenic variants or genes she is an expert in. She can then annotate certain variants to discuss with her colleagues who can also edit this report. Once everyone is satisfied, variants can be selected for reporting, which is the handover point to the multidisciplinary teams meeting or to a medical director. Now logged in as the medical director, Pam, we can navigate to the patient's interactive report and see all the work done by the clinicians. Importantly, Pam can jump straight to the variants for reporting. When satisfied, Pam can add her approver comments, which unlocks the report generation function. The resulting report is fully customizable. In our case here, it contains the discovered causative variant with impact explanation, as well as a summary of the analysis workflows including software version. Once the clinical data has served its purpose, or if a custom bioinformatics analysis is needed, the data can transition into the research environment. Let's follow Bob as he creates a virtual cohort, commissions a trusted research environment that is adequately powered for his analysis, and runs a Jupyter workflow with software he can install. Please note that, depending on the policy, the secure research environment might be fully logged down and no result data or generated images can be downloaded. After Bob logs in, he can navigate to the data portal and create his virtual cohort through a beacon query. Bob wants to study individuals who have cystic fibrosis and also neuroblastoma. He can build this query in the Beacon GUI, as shown, or use large language model support via Ask Beacon. In Bob's case, the dataset contains 5 such individuals which he can now convert into a virtual cohort. The newly created file appears in his My Data section. To analyze this data, he needs to commission a compute instance, and depending on the policy, he can customize his selection or choose from pre-selected options. By default, the user gets dropped into a Jupyter Notebook environment. From here, he can either open a number of pre-configured workflows that the administrator has set up for him to use as a template, or create his own workflow. In Bob's case, he needs to first mount his virtual cohort. To do this, he copies the secure access URL and pastes it into a terminal window. Through this time-limited and otherwise secured portal, the file appears in the Jupyter Notebook environment. Since this is a Python notebook running Conda, Bob can now perform any bioinformatics analysis as if it was a HPC or local computer. Data security and effective handover between clinic and research is managed through role-based access. Besides the clinicians and researcher role, TRECA also has the administrator role, who sets up all the users based on minimal permission principles, and the data manager role, who onboards the data. The system handles genomics and clinical metadata, where sensitive PII information is automatically removed before the data manager moves them into research or clinical projects with their respective security policies. Already supporting real-world genomics initiatives, TRECA helps deliver precision medicine at population scale. Built on principles of trust, sovereignty, and privacy by design, TRECA enables secure collaboration without compromising control. TRECA, today's clinical genomes, tomorrow's advancement of care.